Okay, welcome to our webinar today, everyone. My name's Ron. I'm the director of editorial here at JoVE. So today we'll be having a webinar hosted by Antara Das at the University of Bangalore. So she and her group published with us on a similar topic in 2021. So today she's going to be, updating us on the technique and kind of comparing and contrasting the related techniques. We will be recording the webinar, so we'll send that out to all the registrants afterwards. And we'll also be having a Q&A towards the end. So feel free to add your questions to the Q&A section down at the bottom, and we will address those towards the end. Okay, so thank you, Antara, for hosting today, and I will leave it to you.
Thanks, Ron. Hi, welcome everybody. Thank you for joining in on a Friday. It's a great pleasure to host this webinar today with JoVE, especially on International Women's Day. My name is Antara Das. I am an assistant professor at Azim Premji University in Bangalore. But the work that I'm going to talk about today was done at UC Irvine in California. So let me begin by asking if any one of you have heard about epilepsy. Do you know of someone with epilepsy, or have you seen somebody having a seizure in real life or in pop culture or in some book reference? If you could just maybe raise your hand or do a show of hand, that would be good. Yeah. So yeah, I can see a lot of people raising their hands. So epilepsy actually is quite common. It's one of the fourth common neurological disorder. It is characterized by seizures, and seizures are defined as uncontrolled bodily movements involving jerks. Epilepsy affects about 65 million people worldwide. It includes about 3.4 million people in the US and about 1.2 million people in India. Epilepsy can be caused by several symptomatic causes, including brain trauma, stroke, injury, or it could be because of idiopathic conditions such as gene mutations. Epilepsy could affect people of all age. It could start in infancy as young as 0 to 3 months old, or it could start in adolescence or in middle age or even old age. About 80% of people with epilepsy live in low or mid-income countries, and among them, 75% of them either are refractory to the current epileptic drugs or do not have access to healthcare because of social taboo or because of the increased cost in accessing care according to their conditions. So what are some of the challenges that we have while we want to study epilepsy and find a cure? Have I moved my slide?
What's that?
Did the slide move?
I think it's on the same one. So it's on the epilepsy. Epilepsy is a common seizure disorder.
Okay, all right. Can you see no absolute cure?
No, I can't see that part.
Okay, all right. Hold on. So it's not advancing. Just give me a second.
Okay, no problem.
Okay, there you go, I think.
There you go.
Yeah,
Yep.
all right. So one of the things to understand about epilepsy is there is no absolute cure. We are able to control or contain the symptoms of epilepsy through drugs or through brain surgeries or through implantation of brain devices, but we do not have an absolute cure. Patients become refractory to anti-epileptic drugs over prolonged use. Also, if they have a very severe seizure condition such as Dravet syndrome, they often develop resistance to drugs, and they constantly have to change the cocktail of drugs, and yet they can stop responding to those drugs. Another thing to note is that even within genetic epilepsy, people having the same genetic mutations, they show a very large clinical variability. in phenotypes. That means patients having the same genotype or the same gene mutation may show very different seizure phenotypes, hence they respond to drugs very differently. When we talk about genetic epilepsy, SCN1A gene is a hotspot for epilepsy. This SCN1A gene in humans, it codes for the alpha subunit of the voltage-gated sodium ion channel, NAV 1.1, and over 1,500 mutations have been mapped onto this gene. If you look at this diagram over here, you can see that each spot, each colored spot is a mutation that has been mapped to a human epilepsy disorder in this particular ion channel. And today I'm going to focus on this particular one, the green dot over there. It is in the second transmembrane domain, second transmembrane segment of the third domain of the ion channel. And this green spot causes an epilepsy disorder called GEPS+. Now, what is GEPS+? It stands for Genetic Epilepsy with Febrile Seizure Plus. It is a mild to moderate seizure disorder, and this particular mutation, K1270T, on which we focused our study, it was discovered in a family in America. It had a lot of affected members in the family, and they showed very different seizure phenotypes. In fact, we had stem cells generated from two brothers, one of them who had the mutation and had the seizure phenotype. The other brother had the mutation but did not have a seizure phenotype, and hence he acted as a perfect control for the stem cell lines that were generated. So GEPS plus is caused by an autosomal dominant mutation in which a lysine residue at 1270 position is converted to threonine. So it's a missense mutation, and in heterozygous condition, we find that patients develop GEPS plus. So in Diane O'Dowd's lab at UC Irvine, we developed a CRISPR mouse model, and we introduced the K1270T mutation in the mouse. The founder mouse was then backcrossed into two backgrounds. One was the B6, the black 6N background. The other one was 129X1 background. These two mouse backgrounds have been used before by other researchers to study genetic epilepsies, and we wanted to see if there were differences in the seizure phenotype because of genetic backgrounds. Now, this is one of the first mouse models of epilepsy that were created using CRISPR, and we find that there were no off-target effects. Here on the right, what I'm showing you is that the wild-type mouse, it has AAA, which codes for lysine, or K amino acid. And in the heterozygous mouse, we have edited it to code for threonine. And this one missense mutation in the SCN1A gene causes epilepsy in humans. So the question was whether this CRISPR mouse model would have the same seizure phenotypes as GEPS plus patients. Now, GEPS plus patients have both febrile seizures and afebrile generalized seizures. So what we found is that through EEG recordings, where we implanted a headset on the mouse and we recorded their brain activity across several days, we found that in control mice, there were no seizures, but in heterozygous mice, which carry one copy of the mutation, there were these high spike, high amplitude spikes, discharges, which had a clear beginning and an end, and they lasted at least more than 15 seconds. They had these seizure episodes intermittently. Interestingly, the homozygous mice, they die prematurely around P25, so postnatal day 25. And it would often be followed, preceded by some spontaneous seizures episodes, and they were mostly dying because of SUDEP, or sudden death in epilepsy. Interestingly, human patients are always found in heterozygous condition. We do not have any GAPS plus human patient with homozygous mutation. And since we found that the mouse pups were dying prematurely, it gives us some hints that humans with homozygous mutation either do not survive the fetal stages or they die really early. But febrile seizures are hallmarks of GAPS plus epilepsy. Febrile seizures are fever-induced seizures, and they typically last for about 6 to 7 years, and they are self-limiting. They go away by the age of 6 to 7. But in some cases, uh, febrile seizures persist into adulthood beyond 7 years of age, and pediatric history of having febrile seizures can make a patient more prone to generalized afebrile seizures in their later life. So febrile seizures are of two types: febrile seizures, which are generally self-limiting, and febrile seizure plus, which can exist or which can persist in adulthood. Now, in a study done in 2017, Zhang et al., they studied about 63 pedigrees of human GAPS plus patients, and they found that among all the different kinds of seizure phenotypes found in GAPS plus patients, febrile seizures and febrile seizure plus were the most abundant. So about 44% of patients had febrile seizures, which were limited to 6 to 7 years of age, and about 27 of them had febrile seizure plus, which means that febrile seizures persisted into adulthood. So we wanted to see if the mouse model that we created had febrile seizures. So how could one induce heat seizures in mice? Now, traditionally, how this was done is a mouse was taken in the plexiglass chamber or a glass chamber, and hot air, jets of hot air was sprayed. And this was typically done sometimes just using a hairdryer. And other times, people have used a heating lamp, and the heating lamp could be clubbed with a temperature controller, so you could ramp up the temperature in a desired fashion. But we found that this method of using hot air, especially, was very unpredictable because the heating temperature could not be controlled, and this method was not very reproducible. Also, the glass would start heating up, so the arena had to be changed every few trials, and it was difficult to keep it consistent. Also, there was no way to monitor the body temperature of the mice under study, and we could not accurately determine at what temperature did the seizure induction happen in the mouse because of the high temperature. And sometimes because they were jumping up and down, because when they're having seizures, they're having this uncontrolled movements, it could be really stressful for the animals. They could also sometimes get injured. Their nails would hit the glass, and sometimes you could see redness around the nails. So it seemed like a very stressful scenario for the mice. So we developed a custom built wooden chamber. So thankfully, one of our collaborators, he was very good with woodwork. He built this by hand in his backyard, Martin. So this chamber is 40 centimeter in length, about 30 centimeter in height, and the lid on the top can be open. So you can introduce the animal, you can put a bedding on the floor, and then you can introduce the animal. The mouse has a rectal probe, so we can monitor the temperature of the mouse continuously throughout the experiment. This chamber is fitted with a temperature controller, and if you can see, there is a thermocouple at the back. So it gives feedback to the temperature controller about the temperature inside the chamber. The vents that you can see, there is a fan fitted at the back of the chamber, so it circulates the air within the wooden chamber. Once we have started recording, because of the clear plexiglass, we can secure a camera outside, and we can video monitor this entire trial. And we can see behavior of the mouse. We can even check for the Racine score of the seizures. So we can determine how severe were the seizures and at what time did the seizure start. So this on the right is the heating protocol. We tried a couple of protocols and we decided that we're gonna first preheat the chamber for about an hour. This was done so that there are no, to minimize the differences in temperatures from the first mouse that was put on the trial to the last mouse, because even though it's a wooden chamber, if you heat it and keep it on for several hours, uh, towards the end, we felt that the mouse that was being tried at the last would be getting more or higher temperatures at the beginning. The initial temperatures would be higher. So we had to switch off the chamber in between if there were long trials. But we always started with a preheated chamber, so there was uniform heating within the chamber, and the fan circulated the air. It gave us some time to standardize and stabilize the temperature inside the chamber. So then we increased during the trial would last for 30 minutes. We would increase the temperature by 5 degrees every 10 minutes. And if the mouse body temperature reached 44 degrees Celsius, we would stop the trial and put the mouse on the cooling pad. So what we found is that the heating protocol was highly reproducible. What I'm showing over here are two graphs. So this is the mean body temperature of mice in the 129x1 background. And on the right, we have the mean body temperature of mice in the black 6 NJ background. And they start around 35 degrees Celsius. Their core body temperature is around 35. And when you put them in the chamber, we are tracking the body temperature change. And you can see that both for the control mice and the experimental mice in both the backgrounds, they increase steadily at a steady rate. And you can track the body temperature change that's happening very minutely over several trials, and it remains almost the same for all the mice that were tested. So this was really encouraging, and it was really important that the heating protocol remain constant for all the mice that were screened in this protocol. What we found is that all the gas flush head mice shown here by the orange bars, all of them, the mutant mice had seizures. They had heat-induced seizures. Um, the control mice in the white background did not have any seizures, and some of the mice in the black 6 background, they had seizures. But if you look at the seizure threshold temperature at which they were seizing, that was significantly higher than the heterozygous mice. So I'm going to show you a small video clip where you're going to first see a wild type mouse not having seizures, and then a het mouse having a seizure. And we are using the same box that I just showed you. So here we are seeing a wild type mouse, and it is about 43 degrees Celsius. Did not have a seizure. Here we have a mutant mouse at 42.6 degrees Celsius, and it's starting to have seizures. So this is a Rasain scale of about 4. It is showing uncontrolled jumping. And you can't see the facial contractions, but when you look very closely and you zoom in, you can even pick up facial contractions, minute features of seizures via the video recording. So we published this protocol with JoVE in 2021, and there is a small anecdote to this. So the two pictures at the bottom is when we did the shooting for the protocol, and you see the videographer is helping Martin with the mic. So what happened is my mentor, Diane, she met with an unfortunate bike accident. She fell from her electric bike. She was rushed to the hospital. She had concussions. She was in and out of the ICU just a few days before this was scheduled. And we almost called this off. And I was supposed to fly back to India few weeks after this, after we filmed this. So I was just thinking we're just gonna cancel this. But then Martin said, let's hold on and let's see if he can come in and do the interview part, which Diane was supposed to do. So here we see Diane, we see Martin doing the interview parts for Diane. And it's really a miracle that we pulled this off on that day. And it was a very, very nice experience. We did the shooting in record time, and it was, I think it was really a team effort on that day because we almost had called this off. So this article is available on JoVE. It's open access. I hope you guys will take a look at it. So this work was done in Diane's lab, and I would like to thank particularly Lisha and Andrew, who were undergrads in the lab, and they sort of were involved in standardizing the protocol with me. Lisha, in fact, did a lot of the seizure behavior screens while we were doing this. Before I wrap up, I wanted to talk a little bit about the advancements that have happened in brain research, especially with respect to neurological disorders. So we could imagine 4 different ways in which people are approaching studies of neurological disorders like epilepsy. So one is the use of gene editing or CRISPR tools to generate genetic animal models that carry the same genetic mutations as the human patients, and then replicate the disease phenotype in the animal models, and then screen for therapies. The other has been stem cell based regenerative um, therapy. And I'm going to talk about how genetic strategies have been used to enhance GABA interneuron transmission in the brain. And that has been useful for diseases which are caused because of hyperexcitability in the brain. Also, brain organoid models are becoming hugely crucial to understand neurological disorders because they replicate a lot of the features, a lot of the complexities of the human disorders. So I wanted to give a shout out to Robert Hunt. He's one of our collaborators from UC Irvine. His lab has been working with traumatic brain injury models in mice. And traumatic brain injury affects about 2 million people in US every year. It is called like a silent killer because a lot of times the disease goes unnoticed till it becomes really pronounced. TBI could lead to cell loss, inflammation in the brain, could cause long-term memory loss, and even epilepsy. So in a series of recent publications, what Bobby's lab has shown is that they have transplanted progenitor cells into the injury site in the TBI model. And they have shown that interneurons can migrate into the brain from the injury site to the injury site and develop new connections with the cells close to the injury site. And as a result of these interneurons making new connections and rewiring the brain circuit, diseases like epilepsies could be prevented because the hyperexcitability which was happening in the brain because of loss of interneurons is now compensated from the transplanted interneurons. And they also showed that these TBI mice, which received these transplanted cells, also have improved memory function compared to control mice, which receive TBI induction but do not get the transplanted interneurons. The other approach has been to study the human brain organoids. And Dr. Jack Parrott from University of Michigan has been one of the pioneers in the field who have developed brain organoid models and used them for developing therapies and newer strategies to treat neurological disorders like epilepsy. So in this, a fibroblast cell from a patient could be reprogrammed into a human pluripotent stem cells, and the stem cells can then be programmed into developing 2D models, neuronal models, or 3D organoid models. And these organoids then have greater structural complexity. They have more neuronal diversity, which sometimes is lacking in a rodent brain model because of its size and because of its genetic composition. And sometimes the rodent brain model may not capture all the complexities of the human brain. So these brain organoid models are now a standard for testing all the drugs, the pharmaceutical chemicals, and they can be really useful for precision therapies because the organoids can be developed from the patient's cells themselves. So they have the same genetic background as the patients. With that, I would like to point to some of the resources that are available. There are resources for patients with epilepsy, with caregivers, for practitioners, and for educators. And we have Epilepsy Day celebrated internationally, which is every year in Feb, the second Monday in Feb. And in India, on 17th November, it's Epilepsy Awareness Day. And with that, I would like to thank all of you for your patience and, and for joining in today. I'll be happy to take any questions.
All right, thank you, Antara, for the excellent presentation. And as you mentioned, if anyone has any questions, feel free to add those to the Q&A. It looks like there's one there already.
Yeah, so anonymous attendee asked, can you tell me what are the temperatures you used? The graph was showing 50, 55, and 60, whereas in the video it was 42.1. Yeah, so the temperatures 50 and 55 is to set the temperature of the box or the chamber, but the internal temperature, the 42 degrees Celsius, what you're seeing is the internal core temperature of the mouse. So we are tracking the internal core temperature of the mouse using a rectal probe, and we are setting the outside temperature of the chamber using the temperature controller. So we standardized this protocol, and we found that if we set the temperature of the box at 50, we can slowly raise the temperature of the mouse at 30, from 35 to 42, 43 degrees Celsius at a rate of about 0.5 degrees per minute. That's the accepted rate at which you can heat up a mouse. So we figured that given the dimensions of our box, if we start at 50 degrees Celsius, we could raise the body temperature of the mouse at a steady rate.
All right, thank you for the question. All right, so it seems to be that's the only question, unless there's any others. Like I did say before, we recorded the webinar, so we'll send this to all the attendees and registrants. And if you think of any other questions after the fact, you know, you can respond to that email and we can answer any questions at that time as well. Also, if you have any questions about JoVE or would like to publish with us, you can certainly reply to that email as well. All right. So yeah, thank you again, Antar, for the excellent presentation. Thank you to all the attendees,
Thanks.
and have a great weekend, everybody.
Thank you.
All right, take care. Bye bye.
Yep.